sometimes you have to get the balance between getting high quality data and then trying to get the joint context. All right. So you can use later. We can use alignment softwares, tools online, so that you can check out the whether your result aligns with this sequence. For information, this sequence, maybe I've mentioned before, is from my fourth year students in the lab. She's writing her final year project reports at the moment. So I asked her to do a cloning DNA project, and I confirmed her insert, and this is her insert. So I don't think it blasts. Some portion will blast quite well, but it's not 100% because it's not the sequence as the original bacteria that you can find from the database. So you can also compare that, first of all, the size, and then after that you can do an alignment comparison afterwards, so that you see a sequence. So I'm going to talk about that in today's lecture. So if you all right, another I asked my student, because I didn't do this myself. The problem is the direction of the sequence, because you have from the same one is from the forward direction and from the reverse direction. If I'm not mistaken, it's from 4, 3. Is it 3 or 4? Yamazaki, do you remember? 4 sequence here and 3 sequence from the reverse, right? To get the answer, yes, it's 4 from the forward and 3 sequence from the reverse. You join that first, and then after that you can join all of them together. All right. So if you want a video for me to show that is possible, maybe I can do it afterwards. But then that should be the hint that you get to solve the problem. Most of you had problems with the sequence, with the direction of the sequence. All right, in our class material, we have today, we're going to talk about bioinformatics databases. I often talk about databases, but I forgot that some of them are not just databases. There are, there's 2 slides with 2 PDFs with same name. So that's my fault. I added some slides. So the one with more slides is the final version. This is 28 page. This is 28 page. I don't know what's happening there. Both of them are 28 page. Let me see.
No.
Because I integrated. Nope, it's not the one. Okay. Oh, looks like it's identical. Oh my god. I put the same file in. All right, no problem. I will update them later, but we'll be using this anyway. Okay, so there's a lot of databases for bioinformatics, but we're going to focus on the one that you'll be using for the beginner parts of some basic works. So, but it's not something very new anymore, since we've been using the gene bank for quite a lot. But there are more than just gene bank, and most of them are integrated at the moment. So bioinformatics, as the name suggests, is quite a common word these days, but it originates from the word biological information. And then this is the flow over here. Especially for me, I'm working with bacteria. So for example, you can get the DNA from the bacteria strain, and then from there, from individual strains, you can analyze the RNA, and from proteins, right? So individuals, because we just focus on the basic ones. And these days, there's a lot, I think, when we have the omics at the back, so we're not just looking at individuals, now we're looking at a lot of other things as well. But let's focus on the basic idea. So we can analyze the RNA from the DNA, and then from there, we know that DNA, RNA, and protein is connected through the central dogma. And then, depending on the protein that it has some activity, or it has like green fluorescent protein, then you can see it's fluorescing green. Then you can tell it's will affect the phenotype as well. All right. So for the case of bacteria, for example, let me see if I can. Right, this middle. Right. So for the case of bacteria, uh, like if you have proteins that will allow the bacteria to withstand or tolerate concentration of antibiotic substances, compounds, and then it will become resistant to the antibiotic compounds, right? So that's a phenotype that we can see. Of course, it works with other organisms, so humans. You have proteins for different types of activities, or you have the lack of protein of different types of activities that will lead to disease, for example, right? So, like a mutation in the hemoglobin protein, and then that will cause anemia, right? And selection and evolution, when we're looking at comparing with other individuals, right? So for the case of bacteria and viruses, we compare uh, different types of or sequence of different organisms, and then we can look at how the evolution process happens. So sequences from different organisms, and then see how they're mutated, and then there's a rate of mutation. You can calculate that, and then we can see how enzymes evolve to become the current enzymes, right? And this is the cycle of biological information that we usually handle. And because of the current technology, next gen sequencing, we have a lot of data now. Too much data that it's very hard to make sense of them. So it's very slow process, painstaking process to make sense of this data, and people are keep producing more and more and more data. Now we have people that is not from the biological background, people from computer science, people from I don't know using tools like AI and so on, try to data mining their way through uh, making new findings, right? To make new findings. So these are all slides because bioinformatics are no longer emerging field. In fact, in most universities now, we try to force bioscience students to do some basic programming so that they can be part of the so-called big data revolution, so that they can use the information from these databases and as part of their study and research work, right? So computational biology has been also a lot of progress, especially trying to understand the structure and trying to look at patterns that we cannot see uh, typically, and it's difficult to do with experiments. We'll do with computational biology. For example, trying to fit molecules into structure of enzymes, and trying to uh, computational biology, for example, try to solve a structure of an enzyme as well. So we'll see that example when we'll see protein data bank afterwards. So learn, generalize, predict, right? Predict, organize, integrate, simulate, engineer, and gene therapy. This is maybe the aim an application, but uh, we will. These are the things that we'll be doing using the data from the from the bioinformatic database. Well, mostly prediction. We will try if you have a new sequence, we will predict first of all what is the function, so we know what are the genes we're working with, and then we can see at the organism level, identify what sequence from which organism, from the protein level and from the proteomes level, their interaction between different proteins. Organize, looking at the functions, and then simulate is much more towards the structural microbiology of things. So for especially for like a disease related medical biotech, so the phenotype will be symptoms. From proteins, we will know the molecular structure, we'll know the biochemical function, and then from DNA, we'll get the genetic information. So I'll just go through this very quickly because I think we'll see examples much more meaningful than just learning the theories. So for sequence analysis, there's a lot of genome data. So genomes are all the total DNA from an organism, and now more and more genome data are available in gene bank. And from there, protein sequence analysis can be done, and open reading frames where we will predict which part will be the gene for the protein. We can also do comparative genomics, which is compare the whole genome from an organism to another organism. People don't do that much with humans, but for other organisms, it's easier to do that. And then protein sequence and family databases. So we'll see from the protein sequence specific motifs, patterns that we can analyze and try to assign a function to them. And then there are structural genomics, like sequences that determine the structure of the genes, for example, terminator, binding sites for different proteins, and then promoters. And large scale integration of data. So these are all combination of the previous function. So the difficult part, let me just skip through. So these are the omics, right? The omics genome. So if you have a lot of genomic DNA, then will be genomics, and then when you're looking at transcriptions, then there's transcriptome, and then proteome, and then now combination with all the activities in biochemistry, you have metabolomics. So we can't run away from comparing them, all of them, because that's how we can make sense of the data that we have. And then these are proteomics. Let me just skip through. So the workflow, usually we will, to understand the function, we have to know the protein sequence, not the DNA sequence, because the DNA sequence have to be transcribed, translated to become protein. And once you get protein, you can classify it with two different types of families, categories of proteins that you can know the function. So this particular example is for prokaryotic organisms, right? So in the gene, you have to remove the intron and then just take the exon. So you have a mature and messenger RNA, and from there you can do the translation. And only after that you get the information about the protein, and then you can do some analysis on the function and structure of the proteins. So eventually we want the protein sequence, which means that usually we have a lot of data from DNA, and we need to do a lot of processing, annotation, like try to identify which is the ORF, and then after identify which inside the ORF, which part is the you need to remove the intron and the exon. So the algorithm for doing this is quite advanced now. However, you still need a lot of computing power, because you need to, uh, especially if you have a much more complex organism, you have a lot of uh, DNA sequence to go through. So from the genome project, we know like different organisms have a lot of different types of proteins. For example, E. coli have about 4,288. I'm not so sure how recent is this data. Mycoplasma, very small bacteria, so four proteins. The yeast have 5,900. You can see the more complex organisms, you have more proteins. But most of the protein functions are unknown. So this is the problem, because translating DNA to protein is straightforward, but identifying the function is not, right? And that involves many years of uh, typical laboratory experimentation, and that's the problem that we have now, right? So we have a lot of hypothetical proteins, which means that we know that it's some sort of protein, but we're not sure of the function, so it's assigned as a hypothetical protein. So if you do an experiment with unknown protein sequences, genes, and then you know it's affecting one way or another, but try to find what it does specifically is quite difficult if you don't have any information. So it is known as hypothetical proteins. Well, for example, here is this is a very old data from 2003, but you can appreciate you have a number of proteins that are known because somebody had done work experimentally on it. So this is like, for example, a hydrolase enzyme because we can put a substrate and the enzymes break the substrate using hydrolysis. So it's a hydrolase enzyme. So you can know that, and people can do some measurement, some biochemical experiment on them, and they know it's an enzyme. And then we have the yellow part characterized by similarity. This is because from the experiment data, we know there are certain regions, there are certain homologies, right, from similarity from the experimented sample that we assume the other data that similar to it will have the same activity. So the first enzyme that we did experiments on were hydrolase, and then if we sequence another organism, which technically we didn't do any experiment on that enzyme, but because the sequence shows some similarity, then it is hydrolase. And then there are unknown genes. So these are the unknown parts are, it's conserved. There's a lot of these genes in different organisms or similar organisms, but nobody knows what did they do, right? And then there's also a big chunk of unknown with no similarity. It looks like a gene, whether it is being expressed or not, we don't know. We have no idea. So in my experience, even until today, when I put a whole genome sequence of my bacteria in, about half is unknown, are unknown genes. So there's still a lot of things. Even for the genes that I am studying, it's not being picked up by the database, and it's still being characterized as hypothetical proteins, right? So the aim is usually to find function. Once we know the function, and then after that we can do further research on them. But doing that involves annotation. But if the quality of the data in the database and the algorithm is not good to detect, sometimes you have to go in and do it manually. So the best annotated protein like Swiss prot, now it's part of UniProt. They call it UniProt, and now this is all integrated. It's linked all together with the NCBI gene bank. So we will see how we can use this to find function in our sequences. So the reason why we study different groups of proteins, objectives like to have updated information, and then to avoid errors and so on. But this is long and tedious work. With the knowns, there's low quality and database have always been updated. So there's also a lot of progress in the database that we have. And let's just skip through this because I will explain as we go through together with some class activity together, and then we will see how to use different types of tools. All right. So this is example how when you annotate genes, you have blocks where the database will identify the genes. You can see the yellow part over here those are conserved with different types of organisms here. But don't worry, we will just look at this, and then the structural as well. All right. So what I want you to do next, let's not waste time here because there's a lot of things that I'm going to show you hands-on, and in no particular order because this is something. Actually, the slide I updated, which I failed to upload, I uploaded the wrong one, has the list of normally the tools that we use. First of all, let's in the sequence material here, and there's a Word file here. Now I have to download it again. Hold on. let's download that. Open a new tab file. All right, let's download. Somehow I put it in Word instead of text, but I hope that's not affecting our activity afterwards. All right, hold on, let me share the whole screen instead of just the browser. The whole desktop. All right, okay. So one normal thing that we do usually, we'll just look at how many letters here so that we know right the amount of letters. I think 5,034, right? 34. And sometimes you get a bit off because sometimes when you copy and paste, you'll get it wrong. So we'll be using the answer from last week to first of all analyze this sequence over here. I saw some of you did blast the whole thing, right? It used from the GenBank data, which is okay, I guess. But what's important is let's go to NCBI. Before that, let's visit the site that we've been looking for the past couple of weeks. So from the NCBI here website, so NCBI stands for National Center for Biotechnology Information. So you can see that they use the word biotechnology rather than bioinformation. So that was the original name. There's a lot of resources that you can use, especially on the left-hand side. There's chemicals and bioassays, which I've tried using before. Maybe because I don't do anything chemistry, but maybe there's something interesting here. All right. Data and software. Then there's DNA and RNA. This is where we'll be focusing most. So when we search for a sequence, we usually from the drop-down menu here, there's a lot of like in conserved domains, right? So those are the places that we'll be utilizing a lot. And then nucleotide, right? That's where we search for what we want. Nucleotide. And there's other abbreviations over here which I don't really understand because they keep adding new things, and you'll be surprised how easy for us to use this website compared to previously where you need to do a lot of things manual. Now they process a lot of the text for us quite. So biosample is where when people, you can have a collection of different data. Namely, if you have a whole genome sequence, right, you submit here. Let's look at example. Let's see E. coli. Let's do example for E. coli. And this is a poor sample because, poor example because there's a lot of. So when you click on a bio project, they will consolidate all the information from DNA. If somebody did some RNA and some protein work, so it's all consolidated in one place. But because E. coli is very common. So the first one, you know the organism, and this is the taxonomy. Bacteria, under bacteria domain, there's proteobacteria, and part under that is gamma protobacteria, and then there's an enterobacteriales. So this one goes, if you move forward, the species, genus, what's up, family, and then order, and all the way up, right? So I have problems saying it in English because I memorized it in Japanese. It's totally different. So from there, now you look at the data that you have, how many gigabases of data that you have here, 4 gigabases and then megabytes of data. You can download that for your own research and so on. So let's, so my student sequence whole genome, and then upload it there, and then the CBI will do the naming all the genes and so on. And other than that, GenBank, which we search for sequences. And then there are other things which is somewhat related, which is slightly different. And then from time to time, they change the name. BLAST for searching, for comparing sequences to the database. And then we have, this is for entry uploading. There's a genome BLAST for genome remapping, and then genome workbench. I haven't tried this one. Integrated application for viewing and analyzing data. There's a primer. Primer 3 is a software that we use to design primers. I didn't recommend using this because I wanted you to learn the basics of designing primers. So once you know the basic, it's easy to make sense of how Primer Blast works. And back screen for removing sequence of vector, which we will be doing next week, I think, when we do cloning. All right. So for today, first of all, apart from this, we have also, I usually search for ORF Finder. All right. Open reading frame is another name for genes. All right. So any region that is in frame between start codon and stop codon will be the open reading frame. And the first step to find genes are using the open reading frame find the ORF finder. All right. So let's copy this sequence here. Let's copy and then let's paste it here. All right. Do we have spaces in between? No, I think no. All right. And then you can set a lot of things, like open reading frame usually will start with ATG because that's from the universal start codon from the codon table. But you can also use alternative. Some organism doesn't just use ATG. For example, bacteria, my bacteria uses GTG as start codon as well. So we can do that. We can click that as, use ATG and also alternative. All right. And then ignore nested. So we don't do that. So let's search, and now you find 71 ORF in the sequence that you just joined last week. All right. So the red bars over here are genes, basically genes identified as genes from the sequence. But the problem is this is only the pattern that was detected by using computer, but in real life, is it actual gene or not? That's the problem, all right? So what you can do is you can clone this into E. coli and see if the E. coli has the activity. But the problem is you have to also predict what kind of activity you have to find for, and that involves different types of experiments to do that, which is not very practical. So you can see, let's look at the result as a whole. You can see there are bars and there are directions, right? Different direction. This is because DNA is double-stranded. So it will show from one direction, and then the opposite direction is on the complementary strand, which is the lowest strand, which is opposite direction. And you can see there are longer bars, all right, longer genes, and then there are shorter genes, all right, and you can click on them both. And here, kindly put numbers to the ORF so that you can afterwards refer it, all right? So the nightmare for most students is now we have to find which one are the genes. So look at, for example, we have ORF 69 over here, and then you have ORF 11. They are different length, and if you put your mouse over ORF 11, you have 1,227 base pair with 400 that will be translated into 408 amino acid, right? So that's a general size of amino acid. But at the same location, right, or the same location, within that ORF 11, there is an ORF 69, which is in the opposite direction on the complementary strand. Now, this one is slightly shorter, 269 compared to that 400 plus amino acid, right? So it could be that both could be a gene. It could be that none of one of them could be gene and one is not, and it could be that both of them are not genes as well. So I'll show you how do you first of all predict what are the outcomes. But before that, this is a very tight segment of DNA, tight as in there's a lot of genes in just 5,000 base pair, and usually bacteria has very compact DNA that the genes are arranged like OR14 here, OR15 here. It's almost back to back if it's actual gene, right? So below you can see this is something that I also want to explain. The database will identify for us where is the location of the start codon and the stop codon, and that the strand, whether it's the plus strand or the negative strand. So the plus strand is ORF11. So let's look at ORF11 here. So from the, at least from this figure, from the left to the right will be the plus strand. So the complementary strand will be the negative strand, right? For example, ORF69 over here will be negative strand because it's on the opposite side, right? And then we have frame. Frame is because every 3 nucleotide will be 1 codon, right? So, but if you take from the second, from the beginning, from the second nucleotide, so you have frame plus frame 2, and then if you take from the third, and then you count 3 base pair onwards, then you have frame 3. So you have 3 different frames, right? Which means, for example, ORF11 and ORF21 is on the same strand, which is plus strand, but they are on the different frame, right? Which means that if you use ORF11, the start codon, ATG, and then you can get a very long gene because after 400, was it after 1,227 nucleotide only, you'll get a stop codon after that. But for the ORF21, the frame is different and the stop codon is also different, and then you get a nested, what we call a nested gene inside the ORF11. So either one could be the right gene, right? Or both might not be a gene. So you can, first of all, to identify which is the gene, right? Let's just search for ORF11 first, okay? And you'll get the amino acid sequence over here. Starts with M over here, and then of ORF format to display. You can do that. So it'll give you a FASTA format all the way straight away here. There's ORF11 there, and then there's sequence of amino acid here. So you can search the database SwissProt over here or different database, but usually we just search SwissProt and then we just Smart Blast. If you click Smart Blast, and then it will find other similar amino acid, right? Other similar amino acid to it, and which some has already been worked on so that we can predict the data. So we have a FAD NAD binding oxidoreductase. So we have that. It's a 409 amino acid, right? So that's looks okay because it hits to something, to resolve something which is at almost 99% coverage with 81%. So it's quite confident that it is a gene that has FAD or NADP binding site on it, right? It's an oxidoreductase. So let's check the ORF 69 just now because it's embedded in ORF11, but it's on the negative strand, right? Negative strand. So we will click that, and let's look at ORF69 and run Smart Blast. I hope it will open a new tab. Yeah, it will open a new tab. So no matches, right? There's no matches here. So which means that ORF69 is not a real gene. So it's not, it doesn't exist. Nobody studied about it. So it's just was just detected as a gene because it contains start codon here and stop codon over here, right? So this is our gene, first gene, and then after that we will look at ORF12, right? After 11, 12. So and then after that we have ORF14, and then we have ORF15 or something like, or maybe ORF9. So those are the real genes, and the reason why I know which one the real genes, because we've gone through this before with my student in the lab, and actually this is part of our cloning work, so we know which part we clone inside, so we know which one. But even with that, you can see how the algorithm will still detect which ones are the genes, and you can actually reduce this because you can do it. You can set. Let's go back. For example, minimum ORF length 75. But if you know, we're not looking for any small proteins, we are looking for big proteins. So you can increase, like, for example, 300, so that it will reduce the amount of the results that you have, so that you don't have to look for 71 ORFs. Now you're looking at only 14 ORFs. So it makes your work much more easy, right? So let's look, for example, 6, but I think 300 is a bit too much. But it's 266, right? 66. And then let's run Smart Plus. Another part is I want to show you that it shows you, well, the previous result already did give us an answer. All right, for example, here you can see this hits to an hypothetical protein, which is people discovered this protein, but they don't know what's the function. So it's registered somewhere. It's a hypothetical protein, which is not very helpful. And then we have this similarity to 2,3-dihydroxyphenyl propionate 1,2-deoxygenase. So this is the correct answer, because this is a protein that I've been studying, and the database is not doing quite well in identifying it. So this is the protein of target, and then I'll see like protocataquate 4,5-deoxygenase and so on, right? So this part, because identical proteins, shows you this is the full length of your search, but the green part shows quite good similarities, all right, high percentage of identity to the target protein, right? So for 2,3-dihydroxy phenylpropionate 1,2-deoxygenase have been studied. There's a publication, you can find that information. You can click on that, and then now from here we have an alignment, right? A comparison. So from sequence, this is query, this is subject, right? So query is the one the one that we put in, which is from our ORF finder here. And we can see the similarity, where are the locations that are similar, and then which location are not similar. And then there are, for example, conserved areas, like for example, this part over here, that's AAA, so that's conserved. And then plus is not conserved, but it's quite the same, right? For example, the first plus we have over here, or second, like the one I see just right here, this part over here, the plus means that it's quite similar amino acid. We have V for valine and I for isoleucine, which is, those are two short peptide, not peptide, amino acid residues, right? And then, so as a rule of thumb, if anything has identity or homo identity of around 30% or 40%, is considered not identity. The conserved region, right? About 30% to 30% is considered more or less the same protein, right? So unlike the DNA, protein, we have 20 amino acid, and those 20 amino acid, some of them have similar characteristics, like hydrophobic amino acid residues. Then we have acidic amino acid residues. We have polar amino acid residues. So that will, even they are not the identical amino acid, but they will behave the same. So that similarity helps maintain the same function. All right. So from here, you can, if you go through that and find, there should be, let me reveal the actual answer. Car basal. This is what, if you've been reading my paper, you can see that, and you can actually make sense. So this is an example. For example, I'm working on a bacteria that degrade this compound called carbazole, and basically the structure of the one that you should get from after you identify which gene is from here, which is like this. All right. So everything face on the same direction. So it's either on the plus strand or the negative strand. So all genes are on a single strand. All right. And then you have the car A, and then you have the car BA, car B, car C, car AC, AD, and so on, right? So what happens if we just blast? Let's go to not finder, but instead we blast here, and we put the whole data here in copy, and then let's nucleotide blast this. Okay, and then let's blast. This will take some time. So the unfortunate thing when you do this in class, and every other students will do, my search result will come a lot slower because everybody's searching as well. So because this is a long one, maybe it takes about 30 seconds or more. So we will blast, and we'll see what comes up from the blast. All right. So here our hard, for example, let's take KA1 instead, because I know this one works. So you can see the similarity is quite good. All right. It's almost 100%. It is 100%, I guess. Yep, 100%, right? And from here, does it shows any function? There's no function, right? So that's the problem. So the reason why we don't blast, because it will just compare DNA sequence, but it doesn't give you information about where is the gene, what protein, and what is the function. So that's why OR Finder is quite useful here, because it basically has the ability to identify which ORF is the gene, and then you can check it database. So that's very useful. For information, 10 years back, it's not like this. So we have to do the ORF, and then after that, we have to copy the information, go to SwissProt and paste it on SwissProt, and then wait for SwissProt, which was very slow. And then after doing for 5 or 6 genes, then you get so many things you've copied and pasted that you lost track of what's happening. So this is a lot easier, and it's everything in the cloud, so it makes things very easy to work with. All right. So if we look at graphics, what happens? There are some new features that you can see. Or so this is the actual data, and it will show you. Oh, this is the answer. So because we have a hit in the database already. So the first one is car A, car BA, car BB, car C, car AC over here. So this is the, I think you can scroll this or you can extend the whole region, right? So inside our data, there's 1, 2, 3, 4, 5. There's 5 genes in total. So compared to the 71 genes you search from initially from the OR Finder, so it's a lot more difficult to, it takes a lot of time to smart blast each one. But if you have a hit in the gene bank database already, so it makes the work easier. So what do we learn from here? You have to do all, right? So you check blast, If you can't find any information on BLAST, you go back to or Finder, and then you search each one. And if you have to go deeper than that, then we will look at how do you search for specific domains and so on in proteins, which is a lot more tedious than just blasting. All right, next activity, which is that's the demonstration that I wanted to do, but what we'll do, which is much more related to the group activity that we'll be doing this week, is also incorporated inside the Word file, which is the activity here. So you've been working with this for the past 2 weeks. You know these are accession numbers. So let's first of all take the accession number here. Oh no, I press Control N instead. Right, Control C, and let's work in NCBI again. Let's close everything. And let's go to nucleotide. Nucleotide. All right, done. Oh, and just Control V, pasting the accession number that we have. So what I'm gonna do now is show you how do we use the multiple alignments to design basically a primer that we can use for detecting the species, all right, that we are testing here. So the problem is my accession number leads you to complete genome, all right, complete genome. So having complete genome data is good, but loading time will take longer. So there's now more than 6 megabytes being downloaded. But I think halfway we can find our data. So we'll be looking for to sequence to design a primer that can sequence the 16S ribosomal RNA for this case is E. coli BL21 goal D3 over here. But while waiting, let's also do the other one because we need multiple alignments, right? Okay, no. Let's see if Google's smart enough. If I just type gene bank with the alignment, will it show? Yo, all right, that gives you answer directly. So the next one will be Salmonella enterica, Cerova typhi. All right. So you can see the common trend already. I'm looking for human pathogens, but not BL21. But this is an example of E. coli. All right, and lastly, let's do the third one as well. All right. So we can do directly from Google apparently, so that makes our work easier. No, you have to put the word gene bank in front. All right. Is it? No, I don't get this. It's not successful this time. Apparently. All right. Then I will just do it the normal way. And the third one is quite simple because I take directly from the 16S. So we want to look for this gene, which you can just Control F, and for we're back at our E. coli page. Let's search for 16S, RR, and A. So not that one, not this one. There will be several genes. Oh, before that, I need to confirm that whether there is a 16S ribosomal RNA gene inside this data. Of course, obviously there should be because we're working with a whole genome. From here, you can go to bioproject, if I'm not mistaken. And from the bioproject, we have nucleotide genome protein sequences per assembly. Not this one. All right, let's go back. It's not the biosol project, it's the biosample. No, probably it's not this one, right? Usually you can find data, if it's a new, this is from 2013. You can find the data of how many 16S ribosomal RNA in the sequence that you're looking at. But even if we can't find it, it's okay. Let's. All right. So we just search 16S ribo and it hits here. And altogether, there's 5 hits in the page. I think there should be, no, the rest are not. This is third one, there's 2. Usually in the bacteria, there'll be about 2, 3, 4 copies of 16S ribosomal RNA. So let's just take the first one. Let's assume that they are all identical. Let's assume, or there's 9. There's more than. Let's take the first one. All right, the first one. So this is this. If you click here, you'll get the sequence of RNA, but we were looking for the gene. So we click on the gene, and then it said no sequence, but we can go to FASTA. That will give you the sequence over here, right? So for some pages like Vibrio cholerae, you can search Vibrio cholerae strain PS4 16S ribosomal gene, and it will give you the ribosomal RNA gene directly. So you don't have to open the whole total genomic data. You can just get here the data. So ribosomal RNA is about 1,500 base pair. So this is correct. It's no problem. So but for the E. coli just now, we have to go through the genomic DNA. We will find 16S ribo there, and then we'll get our data. And this will be around 1,500 base pair as well, right? So this should be the same. So let's just leave it there so we can copy it afterwards. This is already in FASTA format. You can know that FASTA comes with this symbol over here, which I don't really know how to describe it. And you have text letters without the numbers. For the Vibrio, now you have number 1, number 61. It helps you read the sequence quite well, but when you copy paste, you have to remove the numbers, which is quite tedious, right? So what we'll do is, so that's the problem that we have here. We go to FASTA format, and you'll get the version without the numbers, right? Without the numbers. So that helps you eliminate one problem afterwards. Next, we'll go to Salmonella, and we have to do the same because now we have the whole genome data over here. The data is too large. You cannot find anything there. So just type 16S ribo. Let's take the first one, click gene, and then click FASTA. And you'll get, for the example that we'll do in class today, we'll use these 3 genes, and then we will find a consensus, a similarity between these 3 genes, and design a primer that will work for all 3 organisms. All right. For those of you who's already working on this, you know that this is a very simple thing to do. But just for the sake of example in class, we'll do something simple. All right. So as usual, we can use our notepad. All right, can use notepad, and let's copy E. coli data. So I will simplify the header for the FASTA file. Just put it E. coli instead, right? And then we will go to Salmonella. Copy this as well. You can do Control C, Control V, but I'm holding the mic, so I'll use the mouse instead. It's using Control C, Control V is a lifesaver. It's a lot faster. All right, and then finally, Vibrio cholera. All right, okay, and we copy that. So bioinfo is the only field that your professor allow you to do copy paste without being scolded, right? Okay. I think more or less it's about 1,500. Next, we will use multi-alignment. We have a lot of software for multi-alignment, but my multi-alignment tool. But we can go to ebi.ac.uk, cluster omega over here, and the multi-alignment for DNA. You can select your input, DNA, RNA. We do DNA and we paste Control A, Control C, and Control V. But when you do protein, you have to do protein. And I mentioned that because your assignment will be on proteins. And option should be no problem, I think. Let's see if it works. Currently, be patient, right? We just put 3 sequences. It shouldn't be that long. And it's just about 1,500. All right. So for a typical alignment project, right? For this is a Clustal tool. Previously, people used Clustal W, but there's updated version because it's of course online software again. There's a lot of you can use Muscle and other softwares. So we have multiple alignments here. If you see an asterisk, that shows there's a similarity between the 3 samples, right? That we put in, sequences that we put in. And you see there's also spacings, right? There's a hyphen for spaces because the difficulty, the difficult part of aligning is not just aligning everything when it's the same length. Sometimes it's different because the first sequence might be slightly longer, the second sequence might be slightly shorter, but they have gaps in between. And that's the spacers that we have. And when there is a conserved region, there'll be asterisks, all right? And then when there is no conserved region, then there's no asterisks. So from here, from just one look, we can determine where is the variable region, which means that it's not conserved, right? So this is the variable region because there's no similarity. And then there will be no similarity here as well. But most of it are conserved, right? And this is because this is very common. 16S is very highly conserved. So if you were to design a primer that will work for all 3 of this, which is useful if you want to sequence, use it for a sequence primer and so on, you will find the conserved region, right? So similarly, like before, this is a very easy thing to do because it's very conserved here. All right, and the one below here is very conserved as well. So to be able to sequence, but the problem is like the primer we designed, let's say minimum we want 15 base pair, all right, 15 nucleotides of primer. If you go from the back here, you have 1, 2, 3, 4, 5, 6, 7, 8, 9. That's less than 15, right? So you have a problem with here, and you can see that Vibrio is C when E. coli and Salmonella is T. So you have 2 of them is similar, but 1 of them is right, and that cause. If only you can replace this C with a T with this C with a T, then you have conserved region over here. And then you also have a lot of a stretch of conserved region before this CG, but for Vibrio is GC instead. So there's a bit of problem there, right? So to solve this problem, you can get a sequence region, a conserved region from here, and that will give you no headaches because everything's conserved. The problem is when you do that, and you know because you analyze the sequence data, you just lost about, this is 1,530 and this is 1,495. So you lost about 40 base pair, right? So your sequence will be shorter, right? So this information might be important to distinguish Vibrio and E. coli, but you have to go shorter. All right. So how do you go about this? Right? When you design the primaries, you sample to the company will synthesize. All right. So you can have, all right, you can have the primer for forward is this. Let's 1, 2, 3, 4, 5, 6, 7. Let's just take randomly. Let's just assume that's 15 because we're not counting. All right. Okay. Let's put that as a forward primer. Hold on. Forward primer will be that, and your reverse will be the challenging part. It's not that challenging because let's choose here. This is a bit a long stretch, but let's just assume it's like this, right? For example's sake. So this is all right. This is what I hate about using Word. All right. So we have 5' and 3' over here. And let's take note of the changes. For example, after GCT, there will be a T as well, right? So let's go here. GT, all right, I lost GCT, right? There's supposed to be a GCT in the middle. So this C is also sometimes a T, right? Because the E. coli is different, right? So at that location, that C could be a T, right? Let's enlarge that, right? Okay. For example, right? And of course, you can do the other two, which is GGC. It could be GGC, right? Let's find GGC there. So is it this one, GGC? So this one could be a C sometimes, right? And this could be a G sometimes, right? So you have this sequence for your reverse. Of course, you'll do your reverse complement afterwards, which technically means, all right, we did a hard work doing this, but you can ask the company to sequence. You can design basically 2 primers for the back, all right? So the one with the G version or the C version or the T version, and the company can synthesize a combination of G and C at this location so that it works with both with both with different types of DNA. So we have that. So for this case, I think let's look at universal primer, for example, for 16S. 16S. All right, there's answers over here. Okay, no, this is not using variable. All right, you can see over here, the 27F and the 1492R. So these are primers where people normally use for sequencing bacteria. And I think when we copied the earlier ones, it should be more or less the same. AGA, GTTT, there's 3 T's there. So there's 4 T's here. So AGA, GTT, right? So this is the forward primer for most bacteria we use for 16S. So basically we just replicated the forward primer. So there's no problem. The problem will be the reverse side, right? The reverse side, and you can see, for example, at the location 1100 and 1100F, this is SF, all right? There's a Y over here, there's a Y. So we know ATCG, supposed to be ATCG, but somehow it ends up as Y, and then there's an R, right? An R over here. So R is not the common base. So there's also an M, all right? There's an M. So I don't remember this, but you can assign a Y is maybe a G or C, something like that. And you can search that one up in Wikipedia, I think. All right. You can look for that. So they represent an M location could be one of the or three of the four base pairs. But we already know N. N could be all four. So if you have an N in between, you can have a combination of A, T, C, and G. So when you design that primer and you send to the company will synthesize that location as a combination of A, 25% of the primer will contain A, 25% of primer will contain T, 25% will contain C, and 25% will contain G. So it's a combination, right? So those primers are known as degenerative primer. And it is degenerative because you can do PCR and it works, but the result of the PCR might not reflect the actual DNA, right? If that makes sense. All right, I hope so. Okay, so just to summarize, what we did was obtain from whole genome sequences by just control F, and we used from we get 16S ribosomal RNA gene from 3 different organisms from bacteria, and then we use multiple alignment to find a conserved region so that we can design a primer that will work for all 3 DNA samples that we have. And we confirm it with because this is a very famous primer. It's called the universal primer, and it looks the same. Of course, for designing this, we're not done here because we need to somehow all the letters will be. You need to reverse complement this so that you'll get the reverse primer. So this one needs reverse complement. So this is not the answer yet. So the basic rule of designing a reverse primer is still applies over here. All right. And all right. So before we get a bit more confused, there's a lot of other functions that you can use to help you search things in the database. For example, if you just type E. coli 16S ribosomal RNA, you'll get a lot of ribosomal RNA from there. Hold on, let me pause that. You'll get a lot of that, but you have to know what you're looking at. This is a different variants of E. coli. There's an E. coli 0157H7, which is related to what you'll be doing. And then Shigella and so on. So you'll also realize that you're looking for E. coli, but you'll get other things than E. coli, like Shigella, Klebsiella, and so on, right? Those are different organisms. So you can use the menu over here to basically limit your search to a specific organisms. You can if you're looking only for E. coli, you can click E. coli there. And let me see, it's not responding, it's responding now. So you will get only of E. coli, all right? But there's also a problem because now you're looking at 1448, there's 999, this is incomplete, right? Because we know the sequence for 16S is 1500 in general, so this is incomplete, these are partial genes. So we don't want to take that. So if you want to screen through your data more, you can sort out with this different length. For example, you can click here, and when you sort this one out, you'll get, of course, the biggest one will be the whole genome, which you have to click and find 16S inside, which is not that difficult. But if you're working with many organisms, then you have to download each one. Then it'll be very, if you have slow internet, it's going to be not a pleasant experience, right? And other than that, advanced search is also very interesting, but you can see the result over here. This is our keyword, our search, and then you can use the Boolean and or not function to look at different types of organisms. So that's not a major problem. All right. With that, let's look at what you have to do this week. All right, which is I think a bit challenging. I hope it's challenging enough. So let's go to activity, database for bioinformatics. All right, and now you have E. coli 0157 strain that have been identified as an important human pathogen causing bloody diarrhea. All right, hemorrhagic colitis and hemolytic uremic syndrome. That sounds nasty, right? To detect this pathogen even at low levels, low amount, PCR assay-based detection method are effective. That's something weird with that English. So that's what happens when you design the question after watching football. All right. So our target here is the RFBE gene, which is responsible for biosynthesis of the 0157 antigen that can be used as a target for detection. So that's our aim, RFBE gene, right? So the objective is to find an inter and intra-specific homology, which is the similarity, for the RFBE gene using the genes obtained from the gene bank, all right? So you will look for that, but you will also look for that gene in specific E. coli, Salmonella, Mycobacterium, and Vibrio cholera, right? Okay, so I show you how to do that. And then after that, develop a PCR assay method to be used by hospitals in your country, right? In Japan, in Malaysia as well. So this is very common. In fact, maybe the same way how people design primers to detect COVID, right? So the key here is the inter and intra-specific homologies, right? So you have the RFBE gene. So inside the gene, you can see there are similarities or differences which are conserved regions inside the RFB genes of this E. coli, Salmonella, Mycobacterium, and Vibrio cholera. So you use multi-alignment to do that, and then design a good primer for that if you find some conserved, right? And there's also inter-specific homologies, which means that in the open reading frame, the stop codon and the start codon and the stop codon just go beyond that, outside of the stop codon and outside upstream of the start codon, and you have to also use that sequence and align them as well, because sometimes not just the sequence of the genes, but the sequence that is nearby, all right, in vicinity of the genes are also useful for creating homologies, right? To see similarities, and sometimes they align much more better than the gene itself, right? If that makes sense. Or you can also use a primer that is which begins slightly up front of the gene, and which includes part of the start codon as well. Or stop codon, which includes the sequence which is after the stop codon, right? So you can use that, right? So it's not just the genes, right? So to be able to do that, so you don't search for RFBE genes, but you have to look at the gene inside the whole genome data. So look at RFBE gene and what is maybe 100 base pair upstream and 100 base pair downstream of the RFBE genes, right? And using that data, you align. So find that for E. coli, do the same for Salmonella, do the same for Mycobacterium, do the same for Vibrio cholera, and then after that, design the primer, right? And the third one is an open-ended question, because you have to think of how do you use this primer. By saying that, I also include the word primers, because it's not just primer. If you cannot find one set of primer, forward and reverse, maybe you have to design several primers, all right? So the advantage of design several primers is that not just you can detect this O157 antigen, but you can also pinpoint whether the O157 antigen is from E. coli or Salmonella, and so on, right? So you can design several primers. For example, you can have one primer for the forward, and then you have multiple primers for the reverse, and you can design that. Or you can design a primer that have a common forward primer but have different length for the reverse primer, which means that if you produce the PCR product, you have 1000 base pair if it's E. coli, you have 800 base pair if it's Salmonella, you have 600 base pair if it's Mycobacterium, and you have maybe 500 base pair if it's Vibrio. You can do that as well. So that allows you, gives you the ability to, by doing one test, you can also get more information from the test, so you can treat the patient with the appropriate treatment depending on what type of bacteria is infecting the patient, right? So at least that's my intent idea. So using the tools that I've demonstrated before, and of course you can use other tools that you want, because there's a lot of tools can be used from the NCBI, Uniprot, and so on from there, right? So think of a solution to this and in your groups, and hope to see your answers next week, right? Oh, one more thing that one more website that I'm supposed to show that I forgot is a protein database, which I use a lot in my class, but it's not really related to this week's topic, but just for completion, protein data bank. So protein data bank is where you can see the structures for enzymes, right? Because once you get protein and then you want to see the structure of the protein, for example, let's see GFP, for example, green fluorescence protein. So this is the structure of green fluorescent protein, the protein that turns that fluoresces in green color when shine with UV. So you can see the structure. You just click on the 3D view, and you can see this. So this is a bit out of scope from our lecture, but amino acid sequences, all right? Beta strands, large beta sheets, right? We call it. Then there's alpha helix. You can see. And somehow there's another foreign molecule inside, which is not, I think, which is it part of amino acid? It is, maybe. It doesn't look like it. It doesn't look like any amino acid. There's something in the middle, right? So this is also another bioinformatic tool that we use a lot to inspect different structures, so that we can sometimes categorize unknown proteins based on the structure and see there are similarities and try to find the basis of identifying unknown proteins. Right. So before we end today's lecture, any question? Any question from the UPM side? No question? There's one in chat. No question. All right. If there's no question, all right, we'll end today's class, and thank you very much for joining again. Please let me know if you have any inquiries. Shoot me an email, and I hope to get you on time. And let me have a check. I think I just got a notification that one student submitted slightly up. I mean, after the deadline, but it's still recorded in my Google Classroom, so it should be okay. All right. So as usual, you can submit through Google Classroom, but I think moving forward we'll be using this Zoom again for next week. So just before next week's class, I will make announcements about what I'll be doing next week. Right. Thank you very much, and I'll see you again next week. So I will stop the recording.